And life has been made somewhat simpler, I think, using programs such as the Phanerate, which is a program that was developed by Steve Creasorn, which essentially goes through the process of taking each of these predicted amino acid sequences and doing pairwise comparisons using BLAST, Clustal, and those programs to establish the degree of relationships, so that genes that have recognizable sequence similarity and therefore have common evolutionary ancestors can be grouped into what we refer to as families. And what's helpful about this is it gives us a different level of thinking about the overall genetic diversity of this particular group of bacteriophages. Again, I emphasize that whilst I'll show you the analysis that has been done with the mycobacteriophages, it is not, I think, a substantially different outcome to what you would find if one were to look at other groups of phages that infect other hosts. So one of the principal conclusions from this type of analysis is that phage genomes are mosaics. They are architecturally mosaic. This is not a new idea. The idea of mosaic genome, phage genome constructions came from a long time ago from doing heteroduplex mapping of early phage DNA samples. But this genome analysis and the comparison of genes and their predicted amino acid sequences, I think, gives us a much finer tuning on how what these mosaics look like and how they're created. And so one of the nice aspects of programs such as the Phanerate is that it enables us to readily produce genome maps where instead of simply writing all of the relatives above a particular gene, one can simply denote what family it is. In this case, it's 860. And the families can be color-coded. And so this is just a slide that shows, I don't know, 1, 2, 3, 4, 5, 6, 7, or whatever, 6, I guess, 6 genomes that happen to be reasonably closely related. They share this organization, so these genes are in the same family. They're related to each other at the sequence level. So you can see these elements of synteny, of these common gene orders. But what's very nice about this display is that you can begin to see those elements of variation and mosaicism. So, for example, there's a gene here. These are related, and they're present in these 2 genomes, but absent from these other ones. And yet, as I've noted here, it's actually found as a gene with a significant level of amino acid sequence identity in some completely different genome. And one can look at and stare at these kinds of representations and see lots and lots of examples of this mosaicism. Whether you look down here, a different size gene in this case relative to here, other genes which are jumping around and are present or absent in these organizations. So this is it's a reasonably old slide, but I think it makes the point that what we see in the mycobacteriophages, but is becoming clear, I think, in the other groups of phages, is that it's quite common to see what appears as genomic mosaicism, where the modules, the mosaic modules, the tiles in the mosaic picture, if you like, seem to correspond to individual genes or very small numbers of genes. I must point out that I told you that the structural genes often form a common order. Sequences may be different, but you see a common and syntenic order of genes that encode the structural proteins. Once you look outside of those structural proteins, the parts of the genome that encode everything else, replication functions, recombination functions, regulatory functions, you see these very high levels of, very high degrees of mosaicism. And so, for example, in this segment of the genome of a phage called Chi8, at the point of making this slide, these were all of the known homologs that were displayed. And so you can see, for example, that gene 87 of Chi8 has a homolog, GP78, in a phage called omega. But the very next gene, 88, does not match omega GP79, which would be the next gene in that genome. But it does match BXZ1 GP19. The very next gene doesn't match GP20 in BXZ1, but it does match CONDOG GP25. So given that the relatives here are telling us something about the evolutionary histories, it's telling us that in this case, each of these genes appears to have a distinct and different evolutionary origin. And so another way of expressing that is by thinking that each of these genes is being moved around by a process of horizontal genetic exchange, where genes are being brought in from lateral means of genetic transfer. And so another nice feature of programs like Famarator is that it provides a representational way of thinking about the evolutionary histories. And so in this case, what I've done is to take 6 consecutive Chi8 genes, and I've called up each of the families that they're members of, FAM 1107, the next one is FAM 780, etc., etc. And the Famarator program provides a simple representation in these family circles of the evolutionary relationships, the phylogenetic relationships. What's helpful about this representation, as opposed to standard phylogenetic trees, is that if we want to compare standard phylogenetic trees, you have to have the same components, the same constituents, the same members. And in this case, the diversity is sufficiently high that you just don't have that. So what's nice about the family circles is not only do you get to show a representation of all the things that are related, but you can also see on the same representation all the genomes that don't have a member of that family. And therefore enables you to simply, at least visually, compare these 6 evolutionary relationships of adjacent genes in the Chi8 genome, which is the guy that's circled down there. If 2 genes are related at the amino acid sequence level, a line is drawn between those genomes, and the thicker the line, the greater the similarity between those. The thinner the line, the more distantly related they are. So I think this is a very clear and I think quite beautiful representation, I guess, of these different evolutionary histories. There are some common themes. You can see these, whatever this guy is here, that genome, and these 2 down here, appear to form as a subset in each of these relationships. And yet the overall relationships are clearly much more complex than that. So this is a more, I think, a more formal way of representing this idea of mosaicism, and the fact that each of these 6 genes has a separate and distinct